Good evening, everyone. Thank you very much for joining this seminar. It's in India. It is after evening, I think evening tea. So it's in evening 7 o'clock. Here it's 1:30 p.m. This topic, discovery and development of new antimicrobial agents from Indian medicinal plants. So to me, it's an important topic because it's linked to COVID therapeutics. So I'll explain the details soon. And also, I'd like to thank the organizers, Green Pharmaceutical Initiative, for inviting me to deliver this seminar. Especially Mr. Shivashanmugam has been in touch with me for a few months, and we discussed. And I'm very much interested to collaborate with them, and then to look at the collaboration opportunities in future. And also during the initial introduction session, it's started with Tirukkural, so it's fantastic. I really impressed with the stanza provided by the Tiruvallur. Agara முதல எழுத்தெல்லாம் ஆதி பகவன் முதற்றே உலகு. It's to my heart. It's really it's very inspiring words from Tiruvallur, and some of the information we took very seriously. Some of the lines, for example, கற்க கசடர கற்பவை கற்றபன் நிற்க அதற்குத்தக. So these lines are very important. Probably some of the listeners, now if they are from other parts of Tamil Nadu, probably they need a translated version, but it's available in Google. Probably you can check it out. But it's these are all very important steps, important information for mainly for students. Some of the listeners, they're all graduate student or postgraduate student, so you can take these information as a stepping stone for your career development. Definitely, it's worth. And also another point I would like to highlight the motivation from Green Pharmaceutical Initiative: desire, hope, and dream. It's fantastic. So during the presentation, I'll highlight how. So how these words helped me to plan and decide some of the work what we have been doing. So to start with, actually today also I'd like to highlight about antimicrobial resistance. So this related to this topic. So that is the issue, and we needed new antimicrobial compounds, and how we could use natural compounds to extract these pharmaceutical products or phytocompounds, and what are the plant-based phytochemicals from Indian medicinal plants which could be used for used in a drug discovery platform. And we have used computational screening method to identify and then identify the compounds. I'll explain the details. How can we do this? And also, I'd like to highlight about one of the research platform we have been working on, which is microbial biosurfactants, and they have antimicrobial potential, and also there are a lot of opportunities for various formulations from those biosurfactant. I'll explain with some concluding remarks. And also, I'd like to tell a little bit about the journey. Actually, during the introduction session, it's very nicely they have explained my graduate, postgraduate, PhD, and until now. And also there is something behind that. So the schools where I started, which is a small village school in Wangal. So to be frank, I started to learn English only after my 12th standard, I would say, because most of the Tamil medium students, those who are listening today, are in some other part of India as the mode of education in regional languages like Hindi or Marathi. So there are various, because I have met some professors in UK, they're from West Bengal, but they studied in Bengali language. Whereas in my case, I studied in Tamil. So literally in my journey from, I can tell you, so it's a tiny village school between 1975 to 87. So after completing my +2 exam, I applied in BSc Zoology degree program at the PSG Arts and Science College. And also that helped to move on to identify the strength of what are the strengths I have to explore. So that helped to move on to the master's and PhD. And also I would like to thank my friends and the network during both my schools and also undergrad program. It's very, very important. I would say those who are listening, if you're studying in undergrad program, probably your friends network, it's very key because I was lucky to have fantastic friends. Um, and also it's personal, but it might help. Uh, Probably in my undergrad first year, I was literally struggling to get through the in the first semester, but I never failed in any of the modules. But I got first class most of the time because of the students' network and also the opportunities to discuss the issues, to learn, and we moved on. So it's important to keep away from any distractions in the first year, and also keep some good friends. And in our case, no, it's not, I'm not telling or overemphasizing, keep away from smoking or drinking or any stuff. So we never had that. So that also helped. I realized that. So now, probably it's about a 30 years journey. In the UK, if we, all these accessibilities are there. But because I learned that to avoid in my schooling days. And also the schooling days we avoided because of our parents. They have insisted keep away from these kind of stuff, and also at bachelor's level. So I could see in the number of attendees now around 100 plus. If you're undergrad student, I would emphasize to focus on your education because that is the key. If you put effort, that will give a lot of opportunities open up, and then you'll explore when you go to the masters and MPhil, PhD. So each stage you'll be able to identify new opportunities, and also what you have learned in the bachelor you can implement, and then it will work towards the career pathway. So that's how, if you look at from 1975 until 2023 now, mostly I spent all my days, most of the either in schools, college, or academic institutions. It's a fantastic journey. I thoroughly enjoyed. So those who are aspiring for a research scientist or academic positions, even for a personal development notes, it's all will help if you have a good friends network from the beginning. And so that I would emphasize to have that in mind. And and also if you have any issues, discuss with your friends and then mentors like your lecturers to get help. And later stage, I don't need to explain. I think during the introduction they have clearly explained. I'd like to tell about Liverpool, where I work now. So I work. This is a beautiful city, and also it's a historical city. We have various museums and a number of so historical buildings, and it's a northern part of England. So the mostly people are very friendly. So I would recommend if any listeners of this talk, if you are planning to do further studies in UK, I lived in both in south part of England and also northern part of England. I would recommend to find somewhere in northern region because northern region mostly it's a friendly atmosphere, and also it's you'll thoroughly enjoy, and it's much more the diversity is very high. Whereas in southern part of England, mainly in London, so you have a lot of diversity. Diversity means people from around the world. You can see people from, I think it's all the countries. So you'll not be feeling like isolated or you're a strange person. So that sort of environment is needed, even for your career development or a work place and so on. So clearly, Liverpool, I could see there is such a huge spectrum of skills, expertise, and also diversity. And that help to identify your skill set for your career program, and also you'll have your personal and social and academic life to maintain, which is very important, and also mental health. For that also, it's important to have someone, um, those who have the diversity background. So to come back to the topic, AMR issue, which is antimicrobial resistance. So clearly, if you look at recent articles, so many antibiotics historically developed, which has been in use in healthcare system, some of them not working. And because of antimicrobial resistant issues, even though they have prescribed and used um, these antibiotics, the death rate associated with the AMR still it's very high. So it's a very very important topic. So we need new and novel antibiotics. Antimicrobial resistance, it's antimicrobial resistant is the ability of microorganisms such as bacteria, fungi, and protozoans to grow despite exposure to antimicrobial substance designed to inhibit their growth. So to simplify this, antibiotics already they have done various clinical trial and it's all well studied and it's implemented, but the resistance developed by the organisms. So even after you take the antibiotics, it's the organisms' numbers are not reduced, and their growth rate. If their growth rate is not reducing, that means the infection and also the impact on human health is very high. So you may need to switch to another antibiotic, and the treatment time is increasing. So it's very important to have these antimicrobial compound should work in the first instance when it's used. And also, I'd like to share about the cause of antibiotic resistance. If you look at that prescribing of antibiotics, probably you might have seen many of the developing countries, including India. Sometimes we might be able to get these antibiotics over the counter in pharmacy. So the accessibility is very high. So the usage also high. So these organisms getting exposed and then they're developing resistance. Then the patients not finishing their treatment, sometimes when they feel better, they stop using the antibiotics. So make sure if the antibiotics prescribed by doctor, so the course for you need to follow 7 days course. So make sure you should complete that course. So that means so you'll be able to completely eliminate that pathogen from the system. So and that will avoid the developing resistant strains from through this practice. And also overuse of antibiotics in livestock and the fish farming. So now these industries they have been using number of antibiotics. So for economic purpose, may need to increase them to avoid any sort of infection to these livestock or a fish, but that also that causes issues around antibiotic resistance. And then the poor infection control in hospitals and clinics. So that is also it's a key issue. If they're not following the procedures, so it's an issue. And the lack of hygiene and the poor sanitation. So this set, I would say pre-COVID, these hygiene and the poor sanitation, the numbers were very high. But after COVID, then from 2020 onwards, probably at the hospital environment, these practices are it's very good, but still they need to follow the protocols very carefully. Then lack of new antibiotics being developed. That is also very important. So this my talk, no, it's related to this. So how the new antibiotics, if we develop that, how it will help. So it's important to avoid to reduce the antibiotic resistance in pathogens. So I'd like to highlight our report, first report. So which is COVID-19, a promising cure for a global panic. So that's we have published this report in April 2020. So probably most of you are listening. You all know about COVID and also the impact associated with that. And if I'm going to ask, please raise your hands if anyone or if loved one or friends or family infected with COVID. Sometimes you might not get that COVID infection. Probably most of us will raise. We have one or two incidences, either from our family or friends. They were affected by COVID. In some cases, COVID related death. So it is an important topic. And during early stage of COVID, so the team from India, South Africa, South Korea, ourselves, and the US. So we did a detailed survey as to how to find out any solution of mainly the antiviral compounds from medicinal plants. We have done the survey and then published the report in April 2020. April 2020, if you recall, most of the countries around the world, we started to feel the pressure from COVID. And by end of this year, completed. So many countries went lockdown and various health impact, economic impact. Most of the businesses shut down and then completely it's changed the way how we live, how we work. So, for example, including the universities and colleges, institutions, all went it's online mode, or and mostly the students were also affected during COVID time because they didn't have much exposure to the laboratory environment and practice and so on. So clearly, it's an important topic, and if you look at some of the points highlighted here, it's implemented in UK, but I think during COVID time, wash your hands, and then second, wear covering. The third point, so make space, distance, and 2 meters away from people. So all of them were implemented, and we have seen the positive impact associated with that. But it's so later that the situation it's relaxed. It's nowadays most of us we are not following these procedure, but COVID is existing. So if you look at the recent report, it's in England. It's a couple of weeks before, so it's weekly death. So this is on 13th of January 2023. So weekly death about 849 deaths per week, and this number it's a trend is increasing. So here we follow various protocols very seriously. For example, even at the university level, we do LFT test kit and so on. It's quite often. Many developing countries, including India, so some of them are not taken very seriously. Sometimes it's a huge population, billions of people, and some of them have access, some of them they don't have access to this. So it's a bit difficult to find out the even COVID infection and the vaccination program. So we in UK we got first vaccine and then booster dose. So now we have completed I think booster dose 4. But even now the vaccine, the COVID infection, it's coming up. Actually, yeah, but I can highlight even last night, even though it's a personal, but it's good. This topic, no, it's important. Last night, my wife called me, and then she said, my wife and daughter they got COVID positive, and she's immunocompromised, and so she has to go to the hospital. So admitted last night, and then discharged around 2 o'clock with the antibiotic IV um, injection, and then they prescribed antiviral drugs. She's taking this morning, and both of them they got a COVID test positive. And my daughter, she got mock exam today in University College of London. It's an important program with mask and with all the health and safety protocol. She doesn't want to stop it. She went and then she's going to attend the test. So now we know how to live, how to go with that, but still existing. That's what I'm trying to tell. Even after 4 doses of vaccination, still we are getting infection, COVID infection. So it's not gone away, it's existing. So a lot of new therapeutics. So that's the area of research we are going to discuss today. It is very, very important topic and a lot of research needed. And countries like India, they have to take very seriously. So already the various programs are going on, but still, if any of the variants from these new variants, if it's um, dangerous than the previous one, then we are in trouble. That's what I would like to highlight. So COVID is not gone away from us. Still, we need to live with that probably at least a decade, I would say, because some countries they don't have access to the vaccine. They rely on some other mode of action. So this topic, research topic, I would like to highlight how we have started. So as I mentioned, we teamed up with the international team during COVID time. So that is clearly an opportunity, I would say, because COVID time, many of us, those who are listening with the academic background, probably you know how difficult it is to go to the lab to set up any lab experiment. So we came up with an idea of, okay, we can't go to the lab. Why don't we do this computational based approach to find out, to screening some novel or new antiviral drugs. So we have used bioinformatics skills to do this. So first we have identified about 605 phytocompounds, mainly from Indian medicinal plants, those who have antiviral and anti-inflammatory activities. So we have shortlisted, and then we have used the binding domain of spike protein. So that sort of approach on SARS-CoV-2 wild type strains under the variants of concerns alpha, beta, gamma, delta, and omicron. But now we have various other variants are available and also many new reports coming up after our report. But still, it's an important topic to understand, and those who have access to bioinformatics with the phytochemistry or pharmaceutical background, probably you will be able to do further research in this topic. It's an interesting area. So our approach was based on extensive molecular docking and the assessment of drug likeliness and the robust molecular dynamic simulations. So mainly we focused on these 4 variants: alpha, beta, gamma, and delta. So during 2020, and then Omicron it in 2021. These are all very important variants in COVID era, I would say. So our methodology, we have used, it's first we prepared the ligand based library. And the refinement of ligand structures. So it's a library of 605 phytocompounds from 13 medicinal plants. Um, we have used the Dr. Duke's phytochemical ethnobotanical database. So I would recommend if you have a botany background or a pharmacy background, probably you may know this. If you want to explore about the compounds or the importance of plants, I would recommend but to access this database, you'll be fascinated to see the information available for your research. It's so that will help to identify some compounds and so a lot of information is available. I would recommend to use it. And then we have prepared a refinement of protein structures. So that's it's mainly software based study. So we use PyMol software, and then molecular docking using Autodoc Vina, it's again another software. Then assessment of drug likeliness using Swiss ADME and PKCSM web servers. So clearly we have used these protocols to identify the compounds and make sure the predicted compounds those initially out of 605, if any compounds, those showed any mutagenic, carcinogenic, cytotoxic, or hepatotoxic effect, we have removed from the list. So that's we looked mostly because when these phytocompounds are considered for the oral dose or hand gel application and so on, make sure it should not create any negative or health impacts. So we have shortlisted based on a positive impact of mainly these um, either mutagenic, carcinogenic, cytotoxic, or hepatotoxic effects. So make sure that compound should work well without creating any impact. Then molecular dynamic simulation. So it provides insight into the conformational stability of protein ligand complex. So we have done, and then finally we have done analyze the using various statistical packages like SPSS software package to find out the significance of these results. And this is the schematic diagram of the information what you've said for the methodology. So those who are listening and if you're planning to do some work, you can use this protocol. And you may need access to a bioinformatician to help you to identify the compounds and so on. So the bioinformatics skills are very important. So this is the website for Dr. Duke's phytochemical ethnobotanical database. So if anyone working on plant-based material, probably, yeah, you can type either the compound or the plants you're working. So then you'll get the information. So it's very interesting database. I would suggest to use that. So in our study, we have shortlisted about 13 medicinal plants from India. Some of them, it's a globally available now. So predominantly, it's all like in tropical plant species, and for example, cumin, turmeric, and tulsi, then garlic, some of the common names now, probably most of us we know. So these plant names under the either tuber or fruits or plant, the leaves. So or yeah, so all of them are the bulbs. It's been used in some of them. It's been used in cooking on a daily basis. We have been using, but the specific phytocompounds from these plants, they have fantastic effect to mainly to develop antiviral compounds against not just COVID, many other the viruses will be able to tackle with some of the natural compounds. Just I would like to highlight this one also. In Government of India, they have the Ministry of Ayush. I think most of you know about it. It's for Ayurveda, Yoga, Naturopathy, Unani, Siddha, and the homeopathy drugs. So it's already they have developed guidelines to practice, and in early 2020, so when our publication appeared related to the natural products from plants or phytocompounds from plants, from Indian medicinal plants. So during that time, if you just to recall, early 2020 in India or in Tamil Nadu, because at the time we didn't have vaccine, we didn't have access to any antiviral compound, but the COVID cases were increasing and the people were very concerned. And there was a huge impact on social, economic impact, and even to go to the next town or village, you'll not be allowed to go. So many challenge, and they have come up with various plans, including to roll out it's immune boost kashayam. It's introduced. I think Tamil Nadu Chief Minister at the time he started, and then it rolled out in the state and many other states. They have used various approaches because vaccine was not there. What are the other way to tackle this issue? Because death rate was increasing, and probably everyone knows, even though the reported death rate was significant, but the government reported death rate is quite low. But and the impact of those death and hospital environment and accessibility to access to the emergency services, all it's a huge nightmare, and a lot of people they were struggling to access the hospital during the time. So Tamil Nadu population about 72 million. So they have rolled out we can use the kashayam to boost our immunity. So that's I think practice. In even in Western countries at the time there was no vaccine, and here we used various medicinal plants combinations. For example, turmeric, pepper, ginger, and boil it for half an hour and concentrate, and then have the dose. So we all, I think many Indian families here they practiced, and we didn't have that much, probably at the time in 2020 or 21 we didn't get any COVID infection. But at the time, we didn't also have any vaccination. So probably it might work. We didn't, but it was not scientifically proven at the time. So just I'd like to highlight. And it's in some other states, some bad practice happened. Like probably I think it's social media, it appeared, and then in various news we heard probably we should avoid in future if anything, because scientifically probably those who studied about microbiology, so clearly various pathogens and also organisms present in these so fecal product from animals, so we should not use it as an edible product or anything, because animals now when they to simplify now, if they are taking a grass in a natural environment, they just eat the grass, but the grass is not cleaned. And they, in the soil, probably those who have a microbiology background, we know there are millions of organisms are there, and millions of
Yeah,
some of them fungus, bacteria. So make sure, since the animals they are not sterilizing while eating, so that will be in the system. So make sure we should not practice any of the bad practices, at least in an educated level now. And we need to create awareness. So clearly, Indian doctors they raise this, and so hopefully these things will not appear again. So come back to our study. So as I mentioned, we have shortlisted number of phytocompounds based on their oral bioavailability and mutagenicity. So it should not be mutagen, it should not be cytotoxic, and it should not be carcinogen, and its hepatotoxicity also should be negative. So, so these are all the the selection criteria we have used, and also we looked. So these phytocompounds make sure it's blocking the attachment of SARS-CoV-2 spike with host ACE2 receptor, and also the phytocompounds inhibiting the cleavage and activation of SARS-CoV-2 spike by host protein, and intracellularly these compounds target against the host pro-inflammatory cytokines. Probably because of time constraint, I don't want to expand this, but it's these are we have selected some logic selecting criteria to inhibitory targets for the phytocompounds. And also we have done interaction profile of phytocompounds with the RBD spike protein of SARS-CoV-2 wild type and various variants. So clearly from the 13 compounds we have shortlisted, so clearly the binding energy scores were very high for abiraterone and rutin. So these two compounds we shortlisted from that work. Rutin, which is available in buckwheat. So if you look at the availability of rutin in root, stem, leaf, flower, and the seed, I think in flower it's about 35 milligrams per gram. It's availability of this rutin in this plant. So that means 35 milligrams per gram. So we'll be able to get around 3.5 kilo per ton. So I think it's a scalable. For example, these kind of, when we identify some phytocompounds, if we want to scale up, And so it's possible, even though they are all seasonal, these plants, it's possible to develop. And another plant to extract abitatrine, we could use wild lavender. So the interaction of abitatrine with RBD spike protein also it's good for both wild type strain and also the variants of concerns. So these findings generate a scope for the future experiments in vitro and in vivo to validate the inhibitory potential of the proposed phytocompounds directed towards safe, effective drug development against COVID-19. So still, as I mentioned, COVID is still existing. Still, there are opportunities for phytocompounds to play. This is mainly for antiviral drug development and so on. Published a paper from this work. So if anyone interested, you can explore all the details associated with this work. So currently, we are working with Liverpool School of Tropical Medicine. So they have a program called Infection Innovation. They have developed a consortium. It's called Infection Innovation Consortium. So that's a combination of academic institution, industries, and various research institutes join and then formed this team. It's a consortium. So they are helping us to do the in vitro testing with our selected compounds, and then they're testing these compounds on SARS-CoV-2 infection, and they're using both wild type strain and also various variants, and they have virology department and to do the so now it's been we have been testing the virus how to find out is it because we got the in computationally we got results shows that the phytocompounds are antiviral against the SARS-CoV-2. So, but we need to validate those results. So we need to do laboratory, we need to have lab confirmation before we develop these, to scale up these compounds and for large scale production. So, and also we are doing toxicity studies, minimum inhibitory concentration studies, and so on for these compounds, including some of the bacterial pathogens, for example, Staph aureus, Streptococcus, Pseudomonas, and mostly until now, they're all challenging organisms, challenging bacteria from hospital acquired infections and hospital environment. So it's a big challenge from these um, bacterial strains. So we are doing and further research with the Liverpool School of Tropical Medicine to find out whether it's possible to use these phytocompounds and some other, and also we have used some microbial based compounds. I'm going to explain in detail next few slides about how we have approached microbial um, approach to develop some product for antimicrobial activity. So so I'd like to highlight this section. It's a biosurfactant, which is a biologically active surfactant or a biosoap, which is predominantly produced by microorganisms. And also it's available because it is a bio, there is also possibility to highlight one plant, which is a Cypindus mucrosi. It's a phytocompound called saponin from Cypindus mucrosi that is a saponin that has a surfactant activity. So that is also coming under biosurfactant group, but it's a seasonal. There are challenges associated because it's a tropical plant. I think most of the listeners from India, they know this. It's in Tamil Nadu, it's used as a shihakai. Shihakai is well-known surfactant in Tamil Nadu because it's been used, practiced for thousands of years, and most of the household they've been using this saponin for hair wash and some of the commercial products available. But I would say for large scale production, to replace entire surfactant with the saponin, probably it's challenging. But if you think about the microbiology platform, I think it is achievable. So I'm going to explain how we can use microbiology platform to achieve surfactant market. So surfactant also have a role to play in antimicrobials because it's a very good antimicrobial agent. Because during the COVID time or even now, we are seeing we have been using various hand gels or hand wash with antimicrobial and antibacterial and so on. So clearly they're all surfactants, some of them chemically synthesized. If it's biologically synthesized from bacteria in large volume, so it's commercially it's possible. As I mentioned, these surfactants, microbial surfactant, so it's secondary metabolites from fermentation. So they reduce surface tension and interfacial tension, and they can solubilize and disperse hydrophobic substance and emulsify number of liquids. So clearly, you might have seen various emulsion for hand wash, but it's in the emulsion. It's that technically it's the combination of surfactant and also hydrophobic substance or hydrocarbons. When we mix it and you vortex it, you'll be able to see this emulsion. It's either oil in water or water in oil kind of emulsion, and we can create nano emulsion, micro emulsion. They have various applications related to to industries. So, for example, for surfactant can be used in personal care, household, pharmaceutical, textiles, institutional cleaning, emulsion in and also various agricultural, including and oil and gas industries. So the quantity of surfactant needed, it's millions of tons. So I'll highlight, is it possible? I have a few more slides to share. And then the cosmetic sector, cosmetics or personal care sector, some of the surfactants, they have the anti-aging property. I'm not going in detail, but just I'd like to highlight lipopeptide, which is one of the biosurfactant produced by Bacillus subtilis. It's a class 1 organism that can be used in the formulations for anti-aging cream. And a lipopeptide from non-pathogenic and non-GM source, mainly from Bacillus subtilis organism. So currently it's used as a lash recovery serum in the local supermarket. It's available. So these kind of products, it's coming up, and it's a bio-based surfactant we can produce using fermentation platform. And a biosurfactant I'd like to highlight, there is a historical trend. So prehistory, if you think about it, mostly natural compounds were used. For example, glycosides or plant extracts are used. Then later, slightly synthetic were introduced about 2000 BC. So that's in the name of soaps. So in the past, mostly we used bar soap or it's a solid soap. Now quite a lot of liquid soaps are available. Most of them, they are liquid soap. Then in 1830s, more synthetic and sulfated plant oils were used. Then sulfonated petroleum hydrocarbons, highly synthetic compounds were introduced in early 1900. So mostly that also linked to the petrochemical industry or oil and gas sector. So that's because one of the byproduct from that clearly fits well with the surfactant need, because as we know, the crude oil process, and so it's very large volume process. So the availability of these compounds are enormous. So completely, it took over the entire surfactant market. So that's why synthetic surfactant available in every industries. But now various research, it's confirmed that they are toxic. Some of them creating very hazardous hazardous compounds from this. So companies or industries are looking for alternate product. And in early 1950s, it's completely synthetic. Mostly it's ethylene oxide or so these polymers were used. Then now in late 1900, so mainly natural. So again, sugar esters or glycoside. So these kind of compounds started to appear. And now, because of sustainable development goals, introduced various initiative and so number of terminologies you might have heard. For example, circular economy, net zero, and global warming, sustainability targets. So all the industries, they're looking for alternate products. So clearly, bio-based surfactants will have a huge role to play, and also we could see the opportunities around this because when we spin out a company in 2024, it's called Teijin Biotech. So we got various funding. So now the companies are interested. So we have been working on that. Some of the product, it's commercially oriented. So and it's not just to one particular country. It's a global requirement. We need it's about 7.1 billion pounds worth of market. So even fraction of this market, if it's replaced with the bio-based surfactant, for example, it's here a personal care market. It's about 940 million pounds worth of market. About you need about 200k kilotons. It's huge volume is needed. Even if you could replace one sector, probably we need about hundreds of large industries to manufacture these compounds or these products. It's a huge sector. So to emphasize some of the discoveries we made, which helped to spin out a company. So it's when I was working at Teesside University, we got a project to isolate novel bacteria from local soil. So we screened about 120 plus strains from various sites, and we screened for biosurfactant production in our labs, and we got two interesting strain. It's two new species, I would say. It's Pseudomonas tecidea and Pseudomonas clementia. So those two helped to spin out a company because it's a novel. There are some novelty associated with the compound, the structure, and also the applications related to these products. And then we scale up the process. We have done phylogenetic tree. Those who have microbiology background, probably they can understand this diagram. And we did a chemical characterization, and also we looked the property of these compounds, and we got funding to scale up. Then from the lab scale, we went to pilot trial scale in our lab, about 7 liter, then scale up to 150 liters with one of the bio-based zero pilot plant facilities in Belgium. And then finally we got a couple of buckets full of material, the product to send out to the customers if they are interested to test in their formulation whether it works or not. Still, I have some compound. Maybe you could see some liquid here. So I have some samples. This is a rhamnolipid type of biosurfactant, and they have mono and dirhamnolipids in the combination. But biosurfactant, it's a big, it's a classification. It's a group glycolipid, lipopeptide, and so on. So another one, it's a lipopeptide. Already I mentioned it's a surfactant or lipopeptide produced by Bacillus subtilis. So another group, it's a sophorolipid. It's produced by yeast. It's Candida bombicola or Stamarella bombicola. So that's the organism it's produced. And also we have done in silico engineering of Pseudomonas metabolism. So to reveal the new biomarkers for increased biosurfactant production. That is very important because normal fermentation will be getting about 7 to 10 grams of a product. But for commercialization, if you increase the yield rate from from the process, that is key because these biofermentation are associated with the nutrient uptake of the organisms. So how to increase the nutrient uptake? So we can do that. So in most of the microbiology lab, we use the process optimization. We need to optimize to find out the best conditions to grow the organisms. We can also do this kind of engineered model approach to look at various pathways and then provide our right kind of nutrient and then follow up how that nutrient is converted into product. Those who have bioprocessing or chemical engineering background, probably you may know the importance of conversion of the feedstock into a product. That's very, very important because if we rely on natural organisms, they use these feedstock or the nutrient for their metabolic pathway to just multiply. And the majority of the nutrient will go through the multiplication of the organisms. And then part of the product will goes to this as a secondary metabolite to produce various product. So make sure that particular secondary metabolite links to the product of our interest. So in this case, rhamnolipid is our target, which is a biosurfactant. So if this organism produce various other products, that's not our interest. We need to focus or divert all the nutrients what we are providing efficiently converted into your products. So that's the objective. So those who are working on systems biology or it's bioinformatics related. So it's possible to do these kind of biosynthetic modeling. It will help when you do the validation of this, but we need to clone, for example, if that particular production linked to a gene, so that gene can be transferred or introduced in easy growing or fast growing organisms like E. coli or Pseudomonas putida and so on. So, and also non-pathogenic organism, then you can scale up. So, and you'll be able to, instead of getting about 10 grams per liter, you might be able to get 100 grams per liter. So you can increase the yield rate. So that is the big advantage of using microbiology platform. But if we are planning to, if we introduce this in plants, it's still it is possible, but you need to grow the plant. So like a seasonal variation and also the limitations, because only in tropical countries in certain season, you'd be able to grow that particular plant. Whereas in microbiology platform, the advantage, you can run these plant throughout the year. If you provide the right conditions, it will grow. And if you optimize the process, you'll get the right yield and you'll be able to commercialize. So, but the operation, the capital investment, it's huge. The return of investment for investors, and we need to look. It's on the business aspects. Probably I'm not touching that today, so that needs to be considered. And then biosurfactant. So it's just I'm checking the time. It's 2:30, so about an hour now. So still we have some time. I'd like to check with the organizers. Is it okay to continue, Dr. Siva, Mr. Siva,
Please.
or some? Okay, thank you.
Yes, please. Yes, please. Carry on.
Oh, yeah. So just to, we have published a report about how we can use biosurfactant for COVID-19 application, mainly to destroy or to denature the virus. I've done some initial work. So there are opportunities to explore. And also currently at the Liverpool John Moores University, we are working on these biosurfactant combined with the chitosan for nanoparticle during nanoparticle synthesis. So that will help to develop, mainly the antimicrobial approach. So this combination of rhamnolipid and chitosan, it's boost their antimicrobial efficiency. So we have been doing this work, and we got some funding to expand that, and we got a PhD student now working on this area. And recently, P&G, Unilever, and yeah, L'Oreal. So all these companies, so it's all international companies, but probably even in India, even in villages in India, we have been using products from these companies, for example, toothpaste or shampoo and so on, or soaps. So they have been investing quite large sum of funds. For example, Unilever, they are invested pledged about 1 billion to reduce climate change impact, and it's in its processes. So the processes, for example, if some companies, if they have been using synthetic surfactant from petrochemical industries for the last 50 years, and they made a few billions revenue at the annual accounts, it's huge. So they are ready to spend that money to find some novel compounds, natural, green, and it's make sure it should not affect it should, and also it will create it's a green credential for the company, so that the people still use these products now without any challenge to the future manufacturing. So these Unilever again, they have number of products in the market. They committed to spend a lot of money for green products, and L'Oreal, they have invested, and they have supporting various startup companies to accelerate mainly on natural products or green, either it's a green process or completely the product when it's produced it should not affect human health or environmental health. So these are all fantastic initiative, and the companies are investing. So that will be a huge opportunities for graduates and postgraduate student, or someone already set up a startup company and things like that. You can look at these details. That will help to, yeah, to develop some new approach or novel compounds, and you have a large manufacturer to sell your product or to sell your idea or sell your IP to them. They will support your company to expand the manufacturing, or they will help to host your company to establish the product into the as a, the consumer product and so. So already I have mentioned about the market for biosurfactant. I'll skip this slide. And it's when we do entrepreneurship or business-oriented research, there are huge opportunities, for when we develop a product. If there is a commercial um, link attached to that, clearly you'll be recognized, and then so your company or institutions will be promoted through various public initiatives and so on. So we got quite a lot of informations from supports from various industries and local organizations to promote our work. And yeah, I'd like to highlight some bullet points, like a take-home message. So one thing now we can, so everyone can do these things. So I found this information from Harvard website, Harvard Medical School website. So choose a healthy lifestyle. So the first point they mentioned, don't smoke. Second point, eat a diet high in fruits and vegetables. I would like even fibers are also good. It's probably, I think in South India we follow vegetarian based food. That's excellent. So that ticks important boxes needed. And they exercise regularly, maintain a healthy weight. If you drink alcohol, drink only in moderation. But I would suggest please don't drink. Keep away from that. That will help you a lot. And get an adequate sleep. Take steps to avoid infection, such as washing your hands frequently and cooking meats thoroughly. try to minimize stress, keep current with all recommended vaccine, and vaccines prime your immune system to fight off infections before they take hold in your body. The vaccine, it's highlighted, but in some countries, I'm not sure whether booster dose already available. If it's available, I would recommend to have it, but still take these points. It's very important. If you are a graduate student, undergrad, first year, keep good friends, keep away from smoke and alcohol. That will help you a lot. So, even though I'm working nearly 25 years in overseas, I had opportunities, millions of opportunities to do these things. But if we have a principles and we follow that, we we can do something interesting rather than going anything bad. To summarize, so there is a huge potential for development of antimicrobial therapeutics from plants and microbes. And the in silico modeling helps to speed up the process optimization. So clearly, I have shown a couple of reports. They were very useful. One is bacteria-related in silico modeling of Pseudomonas putida, and another approach we have done with the phytocompounds to develop, for antiviral. So please look at those articles. That will help to expand your research. And lab validations are very important. So we are very lucky to have an opportunity or access to Liverpool School of Tropical Medicine. They are one of the top facilities in the world. Their publications, the scientists, those who are working, they are top researchers in the world, and they have facilities for viral, bacterial studies, and it's all the work carried out under standards, the regulatory needs. For example, FDA and so those kind of we have access. If you are a researcher, if you develop some idea, try to expand your network and try to get help to do further studies, mainly in laboratories, those who are recognized, and the results you'll be getting now, make sure that will be valid results, and so that will help to scale up. Thanks for listening, and thanks to my research collaborators, UG, PG, MRes, PhD, and postdoc student. And it's a long journey, but it's still I need to. It's a learning uh, every day. Even if I'm teaching now, when I had a group meeting with students, sometimes I learn. So learning is very important. That will help to improve our knowledge, and also sometimes to disseminate. If you get an opportunity to disseminate, don't miss. So definitely, so in this presentation, so initially the organizers, Green Pharmaceutical Initiative, they highlighted about poster presentation. It's fantastic. So don't, if you're a graduate student or postgraduate, even for PhD, probably try to prepare the poster and then submit and see. So that's an opportunity, and it will help to include that information in your CV. So that ticks many boxes for your career development. So even I'm thinking of suggesting my graduate student to submit at least 2 posters. So thank you very much for the opportunity. Thanks, Mr. Shiva, for inviting me. I am very happy to answer some questions, and I'll stop sharing so that everyone can see. I have one more slide. This is the last one. I think we all highlighted all the information. So I just stop it.
Thank you so much.
Thank you.
It's an excellent presentation. Dear participants, now it is a Q&A session. So it is open to you. It's open for Q&A session.
May I go with the first question?
Yes, please.
So I am taking the privilege of asking the first question. Sir, actually, I was thoroughly interested in right from the start of your webinar. So I was thoroughly amazed by the kind of scaling up what you have done. You've gone through the exact starting from scratch. You've gone through scaling up and going to the industry. I think it's an enormous feat that you have achieved, sir. So my question is, when you were presenting about the antimicrobials, exactly antimicrobial resistance, I was working at IIT Madras, and I was working in a project for Indo-German Center for Sustainability. There we were working with triclosan. I think you would have very well aware of the fact of antimicrobial resistance caused by triclosan. So this triclosan compound is found in most personal care products like hand wash and other, I think, soaps and toothpaste. So I was actually, when I was doing this project, I was amazed to know that the hand wash itself, the triclosan compound, it promotes antimicrobial resistance. So it was like it is a compound in the hand wash. We try to get rid of the germs, but actually it gets back again and it forms antimicrobial resistance. So it comes back to us. So I think, yeah, sir. Triclosan, how is the scope for
That's a
exactly?
that's very good point you mentioned. So triclosan, if we are any of the antimicrobial agent for hand wash, now when we apply, so the concentration of that particular compound, it's reasonably good. So it removes the pathogen or organisms from hand wash or a face wash or whatever the application it's targeted. But when it reaches the natural system, these compounds diluted in very large volume of wastewater, for example. When it reaches to the wastewater stream, finally it's end up in wastewater treatment plant, if they have one. If they don't have wastewater treatment plant in municipalities or cities or yeah, then most likely it will end up in a local stream. So in local stream, it will be diluted. So in diluted means the organisms living there will get, that's a small fractions of this compound, but they started to develop the resistance. So at hand or in the system when we apply, it's a reason because these companies, they have studied, and then based on the study, they get the right concentration in the formulation and then create the formulation to get rid of it. And that's how they have been labeling that 100% antimicrobial or 99% and so on. So
Exactly.
100% I can understand, but if it is 99.9%, if anyone listening here, and it's more than 100 participants here. In microbiology, 99.9% still that 0.01%, it's very, very significant number because the bacterial number in a pinhead will be getting millions. So 1 million, it's a 10 raised to 6, isn't it? If it's a 0.01% still surviving after that hand wash, so 0.01% means how much it is? It's still thousands.
Yeah.
That is in one pinhead. If it is stays on the hand, how many pinheads you can plot on my hand?
It's extremely large surface area. And
It's in billions or trillions. We can put it. So still probably, I don't know. It's very difficult to count. Enormous volume of number of microbes still existing. So unless the company says 100% proven information, still 99.9. Whatever these fractions, it doesn't matter in microbiology because we are handling those numbers. Trillions are very important. To come back to your question, yeah, it's in wastewater stream. It's all diluted. Still, those organisms getting exposed to these compounds in low fractions, so they start to develop. So yeah. So
And
maybe
one
the
more
one.
thing is,
Yeah.
sorry to interrupt you. I noticed that in India, the wastewater treatment plants, they do some basic testing, like the COD, BOD, all the
Yeah.
normal environmental parameters. But I never saw any of our right now at this stage. They are not looking for any pharmaceutical or right now. I think we are evolving in that stage.
Absolutely. I think it's now it's all to me. To be frank, a lot of work the focus towards CO2 emission, the global warming and many things related to the carbon emission and so on. But the most important things, a lot of metals, for example, heavy metals, hazardous metals, very small fractions from mining or local industries released into the wastewater. If it's not properly treated, it end up in probably in the well or river or lake. That is a huge impact it will cause. And also another aspect, as I mentioned, pharmaceutical compounds in small volume, it's all end up, it's a environmental damage leads to health damage. So it's a big awareness campaign needs to be introduced to mainly on this area. It's pharmaceutical or hazardous metals because this area it's almost not. It's a lot of research done some of the in Western countries, but in some other countries it's not. There
So
is not much
recently, I
care.
think you would have heard, Hyderabad has got a lot of pharmaceutical companies, and from the wastewater streams of Hyderabad, they were able to, if I'm correct, they were able to identify ciprofloxacin at the range of around 900 mg from the wastewater streams. So think
900
about,
mg.
yes, yes.
900 mg in how much quantity of water?
I think they have analyzed from around 10 liters. They have taken a sample and they have analyzed it.
It's a very big
So
quantity.
still 90 milligrams per
Exactly,
liter. So
90 to
it's
100.
about at 90 ppm. So you can look at the regulatory regulations around this. It's a we need a big awareness program about this because it's
Far ahead of the
yeah,
quantity permitted.
Yeah, far ahead.
yeah.
It's a very good awareness. See,
Thank
I
you.
can tell you, can I join for us? Just since we are on the topic, I just thought I can share some of my experience. I was living in Hyderabad as an analytical chemist, while I was working as an analytical chemist for a drug company. Out of curiosity, just we took some sample from the one borewell within our factory, and we injected in GC. We found that almost all the solvents what we were using in the factory were seen in that particular injection. So I was really scared, and the factory was extended about 100 acres land, but almost all the borewells were the waters were polluted.
Yeah, absolutely. I
So
totally agree.
it
Actually,
is
it's
a
not
very
only in
scary.
yeah, it's not only in Hyderabad. In Tamil Nadu, we have seen in Tirupur, even in Erode district, and many
Yeah.
places, it's groundwater is contaminated. Because I have studied environmental science for a master's, MPhil, PhD. So that's my core area of research involved in. I've worked with various scientists looking at textile waste or leather waste industries, and also how to solve this issue, common effluent treatment plants, and so on. So the underground water, it's contaminated with hazardous metals or
And
pollutants in many part of the small towns in even in Tamil Nadu. I don't know about the North India. Tamil Nadu generally they are a bit aware about this. Even the cases here, I can't comment about many other northern part. They don't worry about these numbers. They just practice to make, the industry need to survive. That kind of attitude that needs to change. Yeah. Thank you. Thanks, Mr. Siva. It's very important. Maybe it's if you conduct some other session later, I'd be happy to join about environmental concerns associated with the treatments and so on. Thank you.
Thank you. Is there any other question? Yes, we have some 2-3 minutes left out.
Yes, sir. Good evening, sir.
Hello, good evening.
Good evening, sir. I'm Sindhu, pursuing my PhD in chemistry, that is phytochemistry in Avinash Lingam Institute.
Sorry, which university?
Avinash Lingam Institute, Coimbatore, sir.
Yes, okay.
I have two questions, sir. Regarding Swiss ADME software, when we are using a ligand in that software, we are getting the total oral bioavailability.
Okay.
Can we get the oral bioavailability in pertaining to the receptor what we are concerned about? Is it possible, sir, or the total whole bioavailability only we can get in that Swiss ADME software?
Actually, as far as my understanding, we got the whole bioavailability, but I'd be happy to recommend my collaborator who helped to do the that particular part of the study. So I can share. If you send an email to me, I'll
Okay,
share
sir.
his contact. He is a bioinformatician, so he'll be able to direct you if any technical challenges or how to select related to bioavailability. Yeah. Thank you.
Okay, sir. Thank you. Sure, sir. And one more doubt I have, sir. When we are doing autodoc using for one particular glycone, there will be many glycosides. There is a different glycoside forms for an glycone. So does really a glycoside makes difference in its binding affinities, etc.?
Okay, it's again the same guy. He will help you.
Okay.
Because I'm not a bioinformatician, to be frank. As I'm a microbiologist, but during
Okay, sir.
COVID time, it's a great opportunity to work with some fascinating scientists. And some of them, they are from India, but working in South Korea, South Africa, and the US. So those scientists, they are very well experienced. So I think I'll recommend his details. Then you can discuss.
Okay.
Since you are doing PhD, it'll be very useful because
Okay,
yeah.
sir.
Thank you, Sindhu. Yeah.
Thank you, sir. Thanks a lot.
Any other questions? Any other questions from any participants?
Good evening, sir.
Uh,
Sir, I too have the same doubt regarding the docking study, sir. Like if we use BioVia drug Schrodinger software, sir, we can validate the protein, but in Autodoc we can't validate the protein. So how we can overcome this one, sir?
I'm sorry. So, thanks, Akshay. So again,
Yes, sir.
probably the right person would be Dr. Ayan Gupta, who helped me. He's a bioinformatician, and I would recommend. Yeah, I'll send his contact details. So if you
Okay.
send your email,
Yes, sir.
you and Sindhu, then probably I'll forward your email with my suggestion, with my recommendation. So you will be able to get some
I
feedback.
can
And
share the email ID of Dr. Raghuvan. You can take from us.
yes,
Okay,
thank
sir.
you. Thanks, Mr.
Thank you, sir.
any other questions? Apart from in silico modeling, any other questions which is related to microbial or microorganisms, biosurfactants, etc.? I have one question. Any product have you taken to the next step, that is from bench to the market side, getting a regulatory affair approval?
Thank you. Thanks, Ms. Sabah.
Yeah, UK. Yeah.
Yeah, so thank you. That's very important question. So currently we have been, we have scaled up two biosurfactant. One is rhamnolipid, another one, surfactant. So currently the work for antimicrobial, antiviral studies we have been doing. So the plan is to scale up, and we are completing the MIC studies. And yeah, we didn't go through the regulatory approval process, but we are in the we have access. For example, in the UK, we have access to large biomanufacturing facilities or contract manufacturers. So they will provide the complete package support. For example, to scale up from 150 liters to 5,000 liter, up to 5,000 liter scale. And also they have a team and the regulatory approval and so on. So yeah, we have good contacts, good links with local industries. And in the UK, probably most of you may know, R&D, government investment for R&D is it's very high. If any research, if it is linked to knowledge transfer or commercialization, so still you'll be able to get a lot of support, both yeah, from local companies and also national, international, and marketing and so on. It's a huge. So there are plenty of opportunities. I'm not sure about India, but I know there are so much of developments happening in Tamil Nadu. They are also providing for startup companies support packages, and yeah, so there are so many unicorns. So that's what the terminology I've been hearing from many companies in India. They're become with various targets, something like $1 trillion economy and so on. It's all fantastic. And also they have to spend money on environmental healthcare, environment, and so on. So that clearly that's that's needed, because here maybe it's here the healthcare system. No, it's a national health service. So that means government take care of us. If we pay the tax, so after retirement until you live, government take care of everything for healthcare, and it's a uniform for all of us. So there is no variation. The private practices are there, but national health system that is sufficient for anyone. I think that's also linked to their life expectancy and so on. It's very high here. I'm not sure about everyone will have that, but it's good. We have that system. So something like that, if it's available in many countries, that will help you a lot for mainly to access to the good healthcare, and also environmental regulations. Make sure the companies and the industries operate without any greediness. Make sure they have to take care of people and the environment, not just the revenue-based approach. Here in UK, partly the government have the scheme. If the company is spending for environmental related or anything, they will get tax credit system. So whatever the money they will spend, probably they will get back. So something like that, if it's introduced, the companies will be interested to spend for environmental care or this kind of scheme. Mostly environmental care may not be able to practice because of very expensive, and there is no return of investment for whatever they make. So that means who will offer, who can offer, who will take care of it? That's clearly it's that's need to go through, but go through in some very good, bigger way of thought process. Then only, I think today they have released the budget. I'm sorry, I'm not going to discuss about it, but these kind of, the big allocation should go for R&D or how healthcare and so on, so that it will make our life bit safe and easy. Thank you.